Hello everyone. I'm your educator Priyanka. My qualification is MSc B.Ed and CSI MNet. Today I'm going to Today I'm going to explain a very interesting topic of cell biology, that is cell cycle and mitosis. Whenever we hear a name mitosis, we understand that it means cell division. But only cell division is not right word to say for cell cycle. Cell cycle also comprises the events and phases before cell division. It means how one cell prepares itself for mitosis or meiosis is also included in cell cycle. So, cell cycle is an event in which a mature cell passes through different phases of cell cycle before cell division, and it also includes mitosis or meiosis. It means cell cycle is a whole process right from starting of preparation phase and mitosis phase and cytokinesis phase, and when a cell divide into two daughter cells, it will all the process starting from preparation to the cell division making two daughter cell will be included into cell cycle. Cell cycle has two phases, interphase and M phase. M phase is mitosis and cytokinesis, and interphase is preparatory phase. For our convenience of understanding, we have divided cell cycle into two phases, interphase and M phase. Interphase is again divided into three phases, G1 phase, S phase, G2 phase. We have to understand that G1 phase, S phase, G2 phase, M phase, all are phases of cell cycle, and they will come when cell will prepare itself for cell division and divide itself into two daughter cells. But before that, when any cell doesn't divide it, that is not going to cell division, it remains in G0 phase. G0 phase doesn't come inside cell cycle because it's a resting phase or quiescence phase. Why? Because for cell division, any cell needs a stimulus for cell division. Whenever it gets stimulus from various sources for cell to divide, then it starts and prepare itself for division. When it will not have any stimulus, it is not going to divide, it remains in G0, that is a resting period. Resting period doesn't mean that it will not do anything. It will do its regular work, housekeeping work, but it is not going to do the process related with cell division. It will do its specific and housekeeping work for everyday basis, but when it will get this stimulus for cell division, then it will enter into interphase. And interphase is also divided into three phases, that is G1 phase, S phase and G2 phase. So I am showing it here. This is G0 phase. This is G0 phase. G0 phase means cell is in a resting phase. Resting phase doesn't mean that it's not doing anything. It's doing all its work for everyday work and its specific work. For example, if a cell of liver, so it will do all the work for gene synthesis, protein synthesis, and other molecule synthesis, which is required a liver cell to do. But it will not divide. It only it will not do the work of division. Rest, it will do all the other work. So resting phase only means that it is not dividing. So, but when it will get stimulus from body of organism or any other sources to go to division, then it will enter into interphase state of G1. then whenever it will get stimulus from outside, it will or inside, it will enter into G1 phase of interphase. So interphase is divided into 3 phases, G1, S and G2, and whenever a cell is getting stimulated to divide, it will enter into G1 phases. I also have written the short name and explanatory name for G1 phase, resting phase, G1 growth and metabolism, S phase DNA replication, G2 growth of structural elements, and is mitosis. So you should be clear about what a cell does in G0 phase and what it does, why it enter into G1 phase. It enter into G1 phase only it gets stimulated by its environment for cell division. So what is, now we will know what is G1 phase. G1 phase, for human cells, usually one cell cycle comprises 24 hours. It's the average. Exceptions are there, and it may take more hour, and it may take less hour also in some certain conditions, but for average cells the G1 phase takes 6 to 12 hours. Whole cell cycle comprises of 24 hours, and from 24 hour only G1 phase takes 6 to 12 hours. So it's the longest phase of cell cycle. G1 is the longest phase of the cell cycle because it takes 6 to 12 hours. During this phase, cell grows, becomes large, make all the protein and different molecules which will be needed during cell division. Cell division is very much regulated and controlled process because it's necessary for survival of the organism. If it's not getting controlled and regulated, then it may cause cancer, because unregulated cell division is cancer. So it has to prepare itself, it has to make many molecules, proteins and enzymes which will regulate the process, and it will also increase in size. If it is required, it will increase its size. So after cell division, the daughter cell will not be small in size. For maintaining size and proportion, it grows itself. It grows the cell physically, and it also copies its organelles, for example mitochondria, ribosome, etc. Now it enters into S phase. S is synthesis phase. And what synthesis? DNA synthesis. In S phase, the copies of DNA is getting doubled. In G1 phase, if you will see nucleus. In interphase, if it's a cell and it's a nucleus, in interphase all the DNA molecule will be very diffused manner. You cannot see. If you see it with electron microscope, all the DNA molecules will be lying inside nucleus in very diffuse. You cannot see it like a chromosome. But for our understanding, I am making it here. So let's see. This is a chromosome in G1 phase. This is a chromosome in G1. Chromosome, and one double strand DNA will lie inside one chromosome. You know that inside a chromosome double strand DNA is present. But when it will enter into S phase, what happens that it will double its DNA content. It means that if it's a cell and it's inside cell there is a nucleus, inside nucleus there is a chromosomes lying inside it, and inside a chromosome there is a double strand DNA. But when in S phase it will synthesize one more strand of DNA. It means that this double strand DNA will open and it will synthesize one daughter chromatid or one more double strand of DNA. It means that it is going to be like this. It will synthesize one more strand of DNA, or it means it is synthesizing sister chromatid, one more chromatid. It is for your understanding how S phase of synthesis phase of interphase is going to increase its DNA content. When you see in the actual cell, you will not find it as a chromatid, because this structure of chromatid only become very visible in prophase, not in S phase. But for your understanding how it will synthesize, you have seen in G1 phase it was single chromatid chromosome, but in S phase it is double chromatid chromosome. Both will be called as sister chromatids. These chromatids will be called as sister chromatids. It means that content of DNA is doubling here. If it's any organism which is diploid, the DNA content is 2n. For a diploid organism like a human being, DNA content is 2n, and we can show it by content of DNA by 2c. When it is haploid, it will be written as C1c. For haploid organism, content of DNA is 1c. For diploid organism, content of DNA is 2c. So in G1 phase it is 2c, but in S phase it is becoming double. So the chromosome number is 2n is equal to same. If, for example, if it's a human cell, then 2n is equal to 46 and content is equal to 2c, n is equal to 23. And but in S phase, here 2n is equal to 46. Chromosome number is not going to change. Here you can see the new sister chromatid is forming, not a whole chromosome. There is difference between a chromosome and chromatid. You can see both the chromatids are joined together at the centromere or kinetochore. They are not separate chromosomes. They are sister chromatids in one chromosome. So number of chromosome is not increasing. It's 2n here also, but the content is increasing. Content is doubling. It means the DNA content is going to be 4c. It was 2c in G1 phase. It is 4c in S phase. So I am explaining it for your understanding what is happening actually in S phase. What is difference between G1 and S phase? It's very important because it is asked. This question is very important. It comes in various examinations like CSIR, NET, and it's very important concept to understand. So in G2 phase, what happens in G2 phase? This phase also associated with cell growth, protein synthesis and organelle synthesis. This phase normally has duration of 3 to 4 hours. From G2 phase, cell enters in M phase. See in this picture, I have shown a picture where you can see number of cells analyzed on x-axis and DNA fluorescence per cell on y-axis. What it means? It means that when DNA content is going to increase, the fluorescence amount is going to increase. This picture is explaining when a cell is going to cell division, or this is the picture explaining interphase state of cell cycle. In the first, you are saying that cells are in G1 phase. This peak is for G1 phase. And chromosome content or DNA fluorescence, it means when DNA is going to increase, fluorescence is going to increase. So you are seeing here 2C. 2C means DNA content is deployed, and 2C content is 2C, and fluorescence is this much. Fluorescence is this much. And then cell is going into S phase of cell cycle, and fluorescence is increasing. And when the cell is going to G2 plus M phase, means till M phase fluorescence is increasing. Why? Because now in G2 and M phase DNA content is 4C. Till G2 and M phase DNA content is 4C. So fluorescence is increasing from 2C to 4C. But after that fluorescence is started decreasing. Why? Because after in M phase, when nucleus will divide and daughter nuclei will be formed, both will have 2N and 2N. So the fluorescence will again go to its previous 2N phase. It means from here it will be like here. That's why the curve is going from here to here increasing, but then it started decreasing. It means that here this diagram is explaining that from G1 to S phase, and from S phase to G2 plus M phase, chromosome content is increasing. In S phase there is a formation of double the daughter DNA. There is the doubling of DNA. Till G2 plus M phase, this DNA content is 4C. After M phase, when the nucleus will divide, the content is again becoming 2N or 2C. So this picture is explaining what happens in S phase, what happens in G1, and till when the content is 4C. This picture is explaining about content of DNA, how it is increasing and till which phase it is increasing and when it will become normal. So it's very now cell will enter in M phase. What is M phase? M phase is mitotic phase. It is divided into two different stages. Mitosis means nuclear division, and cytokinesis it means cytoplasm division. M phase has 1 hour duration. It's the very smallest phase of the cell cycle. And M phase is divided into 5 phases: prophase, metaphase, anaphase, and telophase. Here is one question asked in CSIR Net December 2017. To assess the impact of a newly identified drug when added to a culture of subconfluent HeLa cells, a researcher analyzes the fluorescence activated cell sorting profile of untreated drug versus treated drug. Here two diagrams are given. Here what researcher did, he took a picture of a normal cell, which I have shown it in my previous diagram. Here cell is dividing, it's in G1 phase it's 2N, and in G2 phase it's 4N. What I have shown it by 2C and 4C, it is given here like 2N and 4N. It's the same thing. But when the researcher is giving a drug, what happens is that there is only one peak, 4N, not 2N. What it means, what you can say by observing this picture that is given in this options. So you can, based on the FACS profile shown above, this drug inhibits. What this drug does, after when you will watch this both the diagrams, what you can interpret from that, that's what the question is asking. G1, the drug inhibits G1 phase of the cell, S phase of the cell, G2 M phase of the cells, G0 phase of cell cycle. Which phase of cell cycle is going to be inhibited by this drug? What this drug do inside the cell cycle? You have to answer. Please give me answer in my comment box, and I will give the answer in my comment. This is for your practicing your understanding. If you understand this, it means that you have understand cell cycle. If you not, then try to read and try to answer. Here all the phases of mitosis are given. Interphase, I have already have explained it. What else happens in interphase? The nucleolus and the nuclear envelope are distinct, and the chromosomes are in the form of thread-like chromatin. I have explained it. But what happens after interphase in M phase? M phase is divided into 5 phases: prophase, metaphase, anaphase, telophase, cytokinesis. What happens? The chromosomes appear condensed. They become like real chromosome. Before that they are like a noodles in a bowl in the interphase, but in the prophase they becoming thick and condensed and starts looking like chromosome. And the nuclear envelope is not apparent, and in nuclear envelope become invisible, started disappearing. How? Because there are laminin proteins in the nuclear envelope, and at the time of cell division, laminin protein becomes phosphorylated, and it starts break down the nuclear envelope. And one more thing is happening here: the centrosome starts dividing into two centrioles, and then they started going to opposite poles. In metaphase, thick coiled chromosomes, each with two chromatids, are lined up on the metaphase plate. What I told you, that in S phase of cell cycle, the one chromosome become from one chromatid to two chromatid with sister chromatids. That's what represented here on the metaphase plate. On the metaphase plate, the all the sister chromatids, all the chromosomes with sister chromatids arrange themselves, and the microtubular spindle rays starts connecting themselves with the kinetochore of the chromosome. One chromosome with two sister chromatids, when from where they joined itself, it's called kinetochore. Kinetochore. And this kinetochore is joined with the microtubular rays coming from the centrioles. They joined itself on the kinetochore of chromosome. And in the anaphase, the chromatids of each chromosome have separated and are moving towards the poles. And this microtubular structure and rays, they will separate the sister chromatids, and they take them to the opposite poles. You can see here the opposite forces is pulling chromatids from each other. In telophase, the chromosomes are at the poles and are becoming more diffuse. Now they are becoming again going to the what was their structure in interphase. Diffuse, they become diffuse. From condensed, they are now becoming diffuse. The nuclear envelope is reforming. Now nuclear envelope is again started forming by the dephosphorylation of laminins. The cytoplasm cytoplasm starts dividing. The division of cytoplasm is called cytokinesis. So what happens in the cytokinesis in an animal cell? What happens that one constriction starts from the cell wall, and it deepens inside the cell. You can see here in my next slide how in animal cells how cell cytokinesis is happening. In plant cell it's a little bit different. In animal cell it is happens by, in case of animals, outer layers are more flexible due to absence of cell wall. In such cases a circular constriction appears at equator, and it converges on all sides, finally separating two daughter cells. A constriction will appear. Constrictions means a deep narrow furrow, and it will start pinching off the cells into two, and this the actin protein will help in the cytokinesis or separation of one cell into 10 cells. You should remember the actin protein. This plays a very important function in the cytokinesis, actin protein. But in the plant cell it's different. A cell plate started forming from the center of the cell and it will go towards the periphery. In the animal cell it starts from periphery to center, but in plant cell it starts from center to the periphery and the cell will divide. Some important information about cell cycle. Some cells of human body divide every day. For example, skin cell and GIT, gastrointestinal tract cells. These are called labile cells. Some stable cells of body remain in G0 stage, but when needed they can divide. For example, liver cells, nephrons, pancreatic cells. Neuron cells never divide. They stay in G0 phase forever. Embryonic cells have only two phases in their cell cycle, S and M phase. They don't have G1 and G2 phase because when cleavage is happening after formation of zygote, the cell have to divide very rapidly and they don't need to grow because there is no space inside the zygote. So they escape G1 and G2 phase and they only do synthesis of DNA and M phase or mitosis. So it's very important point you should always understand because it's asked in many questions examination. These cells do not have G1 G2. Mitosis is also called equational. You should also learn remember this equational division, because when you will learn difference between mitosis and meiosis, there is also this question is asked very often. Because why it is called equational division? Because sister chromatids from one chromosome are going to two daughter cells. It means every daughter cells have same number and same type of chromosome. That's why it is called as equational division. MCQ practice based on topic. I have written two questions here. You should pause this video and read this and try to answer these questions by yourself. The major events of mitotic prophase include all of the following except chromosome coiling, DNA replication, nuclear envelope breakdown, nuclear disaggregation. What does not happen in prophase of mitosis? The DNA replication doesn't happen. It happens in S phase, not prophase. When the nucleus of a frog red blood cells which does not replicate DNA is transplanted into an enucleated frog egg, the egg goes through several cell divisions. What would be the reason? Why it's happened? Because isolated red blood cells nuclei synthesize DNA. The nucleus plays no role in cell division. An enucleated frog egg can divide. The cytoplasm controls nuclear DNA replication. The answer is the D. The cytoplasm controls nuclear. You should read this question, pause this video and try to understand why it is the answer. Then now this was the today's video. Thank you. We will again meet in my next video. Thank you.